But we also see with that, we saw that there was a more selectivity towards some of these shelf life organisms when using PAA. And so that's something too to consider, right? So we've got to start thinking not just pathogens too, which pathogens are very important. We want food safety, but we need to think shelf life too, because our products don't have a very long shelf life, right? We, out of the meats, we're probably one of the shorter shelf life, especially for tray packed foods. So anything that we can get an extra day maybe is very important, not just for us, but for the consumer as well.
Hi, everyone. Welcome to the Poultry Podcast Show. I'm your host, Karen Grogan. And on today's show, we're going to dig into the world of food safety. And my guest today is Dr. Dana Dittoe from the University of Wyoming. Welcome to the show, Dana.
Thank you so much for having me. I'm very excited.
Excellent. Dana, why don't you tell us a little bit about your background? You kind of count in the category of new faculty there at Wyoming, and tell us how you got to where you are in the world of academia.
Yeah, of course. So I have been across the United States at this point, so it's always a fun story to share kind of how I started. I grew up in Delaware, where the chickens outnumber the people, and never really thought that would be the career role that I would go into. But, that's ultimately what God had in store for me. And I decided to pursue an undergraduate degree in poultry science at Mississippi State University and got into undergraduate research there, looking at applied research within poultry litter management and applying in-house composting. Did I think that's where I was going to go for the first researcher project? No, but it led to a lot of opportunity and open doors. So I did that research with Dr. Aaron Keese when he was at Mississippi State, and I actually went on to pursue my master's under him as well. In my master's at Mississippi State, I really focused on pre-harvest food safety. One of the ways that we can target pre-harvest in the poultry industry is really through the diet. So I wanted to really understand if we start feeding organic acids or even a probiotic of lactic acid bacteria, how would those two work? And would they improve maybe our gut morphology, gut health, and then improve the back end at processing? So would we have reduced potential pathogens at processing? It was really great experience and training that prepared me for my PhD that I pursued at the University of Arkansas under the direction of Dr. Stephen Rickey. So I pursued that in cell and molecular biology, which is just fancy terms for I got to pick my classes. So I got to do poultry and food safety, which was really where my passion was that food safety sector. I started with pre-harvest food safety, again, looking at different antimicrobial interventions or in-feed supplements, and then went to post-harvest food safety, which ultimately is what my dissertation was the post-harvest food safety, where I got to look at both organic acids and inorganic acids and how those can be applied to reduce pathogens like Campylobacter and Salmonella and Coli in the processing facility. Also worked with aqueous ozone and a couple other different products like Amplon. So pretty cool stuff that I got to do there that really opened up a lot of doors, both in the industry as well in academia for me. I actually followed Dr. Stephen Rickey when he went to the University of Wisconsin-Madison and pursued my postdoc there. My postdoc there was not just in poultry, really got to branch out to more meat matrices and expand my pre and post-harvest intervention strategies, looking at organic and inorganic acids. And that is ultimately what helped me get my academic role here at the University of Wyoming. So I'm an assistant professor. My title is meat microbiology, which I think is really fitting because it's not meat and poultry. Poultry is meat. So, but I am able to study beef, pork, anything that I want to do as well as poultry. Very excited to be here in my role at the University of Wyoming. They're very supportive of my poultry background and how that can also strengthen what we have at the University of Wyoming. So really what I'm targeting now in my research is trying to make these applications more targeted and understanding, so especially at the post-harvest level, if we're looking at alternatives to PAA, what is that implication? Are we affecting the microbiome differently of that meat matrice that will then impact shelf life, or even maybe help harbor or hinder the harboring of these pathogens like Salmonella and E. coli and Campylobacter, as well as trying to make applicable quantification and differentiation methods so that we can actually accurately say there are 10 cells or 100 cells of Salmonella. So really helping with that too, not trying to reinvent the wheel, but help the industry along and maybe validate even that research that they're doing out there in the industry with some of these products like the Hygiena-Bac system. And then as well, really just trying to improve food safety and understand pathogen survivability in meat matrices. So really excited, but that's kind of a lot about me.
Great.
Not a little bit, but a lot.
That's good. So. That's good.
One of AB Vista's core strategies is to give customers the flexibility to do more with less, which is a common theme among many companies and producers in today's industry. As a science-driven company, AB Vista has proven results to help our customers achieve optimal performance using customized programs with our core phytase and xylanase.
So you're in terms of, meat microbiology, let's talk just super briefly so we know what players we're talking about here. So what organisms are we focused on? And in terms of the sort of performance standards from FSIS, and that framework that we all have to operate under, sort of what those mean, and then how we do, like how we are going to sort of fine tune our detection. So that's a lot of big topics, but we'll start with the bugs.
Yeah. So I normally focus on Salmonella, Campylobacter, and E. coli. With poultry background, primarily that has been Salmonella and Campylobacter, with me working as well in beef, E. coli,
right.
Those shigatoxin are top ones that our adulterants are very important to continue to knock down and understand. They didn't go away. We're just getting better at reducing them. For Salmonella, which was not an adulterant previously, we are hitting a new era, and we are, each person has a different opinion on it. But I know it's going to be very hard for the industry to make some of these changes. So, earlier there was of this year, that the FSS declared they're going to make Salmonella an adulterant in raw breaded stuffed poultry products. So there have been multiple outbreaks, both in the US and Canada, revolving around that since 2017 and 18. So you can educate the consumer. That's been part of the problem, is the consumer's not following directions.
Follow the directions on the box. This is a partially or not cooked. This is a not cooked product. You have to cook it fully. Yes.
It is a raw product. And, with the invention of and having it at home, right? Air fryers, people are making their own directions, are following Google.
Yes,
or whatever they're looking
social
up.
Media, and even just like packaged chicken cooked in an air fryer, you need to check to make sure that it gets to the 165 degrees of fully cooked. But no one mentions that part in their, Instagram post. So
yes, no, just let it cook. It'll be fine. But it's not. And so after relabels, after trying to, validate different cooking techniques, putting different boxes, nothing really worked for that. And so we're still hitting that point. So for a while, or a couple years, limit of detection really was where they'd start pulling off those raw products. So what you would see is, if I believe it was, you could either do 10 or 100 cells was what most of the industry was doing, based on risk of that product. And they would pull that chicken from the product if it exceeded that limit of detection. The problem that we have for detection methods is that it is very difficult to get at one cell. If we're talking just standard PCR, no enrichment, a lot of PCR, which is what's utilized to quantify and detect these pathogens in different meat settings, especially through the USDA, FSIS, what they recommend, a lot of those techniques are hitting between 4 and 9 logs. With enrichment-based detection, you can get down to 1 log. So basic using like the Hygienobacter system, when they use their enrichment-based, you're using log growth, right? That log phase is what you're hoping to do that based off of. You then are able to input your cycle threshold and detect based on that whole linear curve that was generated off that logarithmic growth. So it's kind of a fancy way to put it, but they're basically able to put an equation in, and you get out of that what your initial starting is, because after that enrichment, you have 4 to 9 logs, right? So that's the hope and that's the goal. But we're getting more sophisticated technology and extraction methods that are enabling us to get down to maybe 1 cell. And that's being pushed. I know BioMaru, they've really worked hard, especially on the DNA extraction side. So their detection method, where you're just saying, is Salmonella here or not, has a different extraction method of that DNA than they do when they start to do the quantification. So they changed up their protocol so that they could get that limit of detection, which is really important. And so we're starting to see a more applicable understanding, something that's more real time. We're still waiting to get some of those results a couple hours, right? If we're extracting DNA, doing the cycle. So it's not really in line with perfect production, but we're getting closer to that. We're not waiting 24, we're not waiting 96 hours for a result. We can have that result in maybe 4 to 6 hours. So that's the hope and the dream. There's a lot of movement to move even further beyond that, beyond PCR. But I think right now that's where we're stuck at, and that's where the industry is working on. And it does work. It works well. But you don't. One of my concerns with this whole Salmonella as an adulterant, there's 2,600 serotypes.
Right. I was just about to ask that question. I was like, how do you know that the one you're detecting is a bad player?
Exactly.
Like, we have bad players, and we have, we have like Kentucky. No, we know it's there. It'll test positive, but it's not pathogenic. It's not pathogenic.
Yes.
So,
and it's so persistent in that environment.
It's very sticky. It hangs around. It's hard for us to get rid of, like, no matter what.
Biofilm former. It's creating an environment for its friends, basically.
Okay, so based off of that framework, we have these ones that, we know we have them in poultry production. We know that they're there. Can we get to a detection method that would allow us to detect the ones that are of concern? And that then the test is that specific? Is that technology available?
So there are some like multi-array methods that people have proposed. It's not as, I think we still have probably 5 to 10 years to trying to get that many targets. Most PCR, so most of our technology when we do multiplex is limited to 5. Maybe 6 targets. You might be able to get 7 targets with some more expensive machines. But most of the commercial targets that we can do based on the channels a fluorescence, when we are able to do qPCR, you'll have 5 channels. So you can have one internal control, which normally we do an amplification control to make sure that we're not botching the kit each time, that everything's going right. And then we have our 4 actual targets. So, that's really easy with Campylobacter when you're looking at 3 different, right? Campy, Coli, jejuni.
Yes, you have only 3, not the 2,600 that we potentially have with Salmonella.
Exactly. So, maybe that ends up in the future. Somehow we're able to identify using maybe next generation sequencing and then do qPCR simultaneously. Maybe that's something that we can target towards and doing that quicker. And there are there are sequencing platforms where you can sequence on the spot, but those, they're not going to have large library sets. So you may be doing one genome at a time, right? One bacteria at a time. And that you still have to culture to get that. So we're limited some on that. But I think in the future, it would be possible. There's also other types of instrumentations that could potentially do this. But they're so specific. I'm trying to remember the name of it. I have a friend that her husband works for a company making them, but the equipment is so specific that we would have to have an extensive library. But again, you still have to get back down to a single isolate. And we're not picking single isolates when we go and take a carcass rinse from the plant. We then have to do that whole streaking for isolation multiple times and then send that off. So we're still limited. What we could do is quantification and then maybe do CRISPR serotyping at the end. I know that Nikki Shariat, so brilliant, is most likely working on how to quantify and do CRISPR serotyping at the same time, which would be phenomenal. That's also great technology and helps us to better understand what our populations look like, because they're not consistent from pre-harvest to post-harvest. So, but that could be another solution. Sorry, it was Raman spectrometry is what I was trying to remember. So Raman is very specific. They use it in a lot of drug applications. So if you're a pharmaceutical company, they can pinpoint that or say yay or nay if it's up to the standards of purity. So that's another one. People have started to look at it, but like I said, it's very specific. And again, we'd have to get back down to pure isolates, and that is time consuming. We're talking about 4 to 5 days to have a pure isolate to send off.
Right. And especially, for salmonellas, we typically do, some type of an enrichment or, selective media. And then you get, you pick a colony or 3 or 4, depending on, what's there. And then there's an identification step. So in this framework that we started the conversation with of labeling it as an adulterant, how do we keep business flow, commerce moving if our detection methods maybe those steps get shortened a little bit with some PCR or, real time, it may take a day, things would get, at that last step identification part, but then we're still quantifying. So in terms of adulterant labeling, that you're talking about 0, like the results have to come back completely negative.
Yeah, I think it's like you have to have below 1 cell is the limit of what the actual directive states. So, I mean, you would be having a really hard time trying to keep that flow because as the current setting, like we've talked about, it's not real time. You can, if we're saying it's less than 1 cell, then you could get that answer within 4 to 6 hours. So you might be able to get that, but you can't get all 2,600 serotypes. You won't know that. So, there is, and maybe that is what their thinking was of why to declare it, is we just don't have the technology. But at the same point, if we knew, hey, it's typhimurium infantis, Enteritidis, those are our adulterants. We could target those 3 very easy. Or if we chose the top 5, we could target those.
So in terms of the, sort of the regulatory environment, it's a very specific product that is linked to this. So it's a very, narrow segment of our industry. And however, when we start down a regulatory pathway, the fear is that it's going to expand to all poultry products. And, the industry has made incredible strides in terms of reducing a lot of the things that you talked about and probably done on your research in terms of pre-harvest and post-harvest. I describe it as we have to use all the tools in the toolbox to get to, low levels going into the plant. And then we have to use all of our plant interventions to get it even lower. So do you think that further reductions can occur? And if you had a crystal ball, how would those, how do we continue? Or have we sort of met this sort of biological threshold of sort of how we understand the ecology of, these particular bugs? Like Campy, we don't have a lot of interventions. We don't have things that reduce it. So are we just to where what we can do? Or are there interventions that are left out there that we need, the next brilliant mind to come up with? So
that's actually a really good point. One of the things, you said that we've used all the tools in our toolkit. So multi-hurdle approach is very important. One of the things that I've noticed more recently is we're throwing peracetic acid left and right in the processing facility.
Everybody uses the PAA. Yeah.
And everyone has a different formulation. And I will tell you, they're not all equal. The stabilizers make a big difference based on the product. So there are PAAs that are better than others. We've seen I've had potential research that we have seen that effect. They're not all equal. They also have different hydrogen peroxide concentrations as well. But when we're focusing on one product, right, we're not really doing a multi-hurdle approach. They're really just having the same hurdle in multiple places. And we're applying it in the water, which is, we've got some physical action there. And physical action with water, we normally get a 1 log reduction.
Right. Just the washing of the
carcass.
Just the washing, we get 1 log.
Every time that I have done a study, I have a wash control with water. It is about a 1 log reduction every time. It doesn't matter if it's, we're doing it on a thigh versus a whole carcass or a turkey. It's normally a 1 log reduction. And then you add your chemical and you might get an additional 1 log reduction. I've seen people report 2 log reductions. I have never truly seen that. So it's 2 logs when you compare it to a no treated, no wash control. But you have that physical action plus the chemical, you can get a 2 log reduction. But if we're not changing out the mode of action, right, of the chemical intervention, we can have persistent populations. Maybe that's why we start to see more Salmonella infantis. I mean, it has been seen that not only does it have a mecoplasmid, but there is some notions that it's more acid tolerant organism than its other Salmonella siblings, right? So there's that concern. One of the things that I've really tried to do is PAAs and organic acid. Let's try an inorganic acid, or let us use other products and, different mode of actions, trying to understand that. And that's really something that I'm trying to get into with my research is, okay, what is the phenotypic and genotypic response of Salmonella or Campylobacter to them? Because like you said, Campy doesn't respond really well to anything. I mean, I can look at it the wrong way and I can't get it to grow in the lab.
Right. It doesn't grow. It's incredibly impossible to grow in a lab. Like you have to do all these extra special things to just, tuck it in bed and make it grow. But
special equipment, extra money
it's
spent.
so finicky. Yeah.
Yes.
Yeah.
But then it's persistent.
It's persistent. Foods. Yes.
It likes friends. That's one of the things that we have seen is that normally it's community structure. It likes friends. Even if you're getting onto some of these selective medias, I know I just completely jumped, but it's such an interesting thing that when you do the selective media and you would try to take an isolate from it, and then say you go to take it to multi-type, like get it typed, right?
Yeah.
Molly-top, yeah. It's not camping. And I had a student that was doing a lot of retail level.
Right. Sampling.
He would try and isolate Campylobacter and a couple other different pathogens at the retail level and just different companies. That's just part of the project.
Just as a surveillance kind of what's in the case.
Surveillance project, yes. And one of his issues is he was using all of the different types of media. Can't be CFEX. He's using a chromogenic agar. I got him to start using modified charcoal, so that MCCDA. And then I have him add a few extra antibiotics to it, so it's a little bit more selective for poultry. And still having issues. And then I was like, well, go get a pure culture and let's see if it grows. The pure culture grows fine. So it's,
your culture methods work, just finding it in the meat product is difficult.
Yeah. And especially, one of the other things that we might not be picking it up. I heard, we have a campy negative or no campy and a processing facility. And I was like, that doesn't sound right.
Ew, you just aren't finding it.
Yeah. One of the concerns that I started thinking about is how long are we holding our samples prior to getting it
to the lab?
They hold
on to those things forever.
Yes, and campy is very sensitive. So if I even store it on a plate, I'm not going to store it on a plate for probably more than a week, and even then, I don't trust it. To reuse it to streak onto a new plate or to use it as an inoculation because it doesn't survive very well. It's microphilic. It needs that environment. And so if we're not providing that environment and we're keeping it much colder than it's used to, it's not like Salmonella who can grow literally when you look at it.
Yes. Yeah.
Yes.
Yeah. Everywhere and will grow just here.
Yeah, exactly. You could put it on the countertop and it'll grow, right? We've done multiple studies where we've done that.
They're very different, very, very different, bugs.
All of our methods, all of our enrichments that we have used were based off Salmonella. And a lot of that, especially the buffered peptone water and the neutralized and buffered peptone water, we based that on Salmonella. And now we're expecting similar results in Campy. And we've done work too where we've looked at the selectivity of buffered peptone water and neutralizing buffered peptone water and what this does with enrichments that you're already utilizing based on USDA, FSIS, the Microbiological Laboratory Guidebook, all those methodology. It changes that microbial ecology on that plate completely. And you start to lose Campy when you're looking at Campy enrichments because of that neutralizing buffered peptone water. So that's also something we have to take into account. We may not be picking it up because we may not be using
sampling correctly.
Yes, exactly. And we might be holding samples too long, and buffered peptone water is not going to allow it a stable environment for it to survive too long. So that's a concern, right? But when we go to reduce it, we really have to understand what we're using to reduce it in the processing facility because, right, PAA reduces Salmonella really well, but I haven't seen that same reduction on Campy. Normally across the board, I see maybe a half a log reduction in addition to water, no matter what really antimicrobial we use. It's been pretty consistent, whether it's acidified sodium chloride or if we maybe use the Safe Foods, their CPC, or if we use PAN. It's very consistent across that board. And maybe that's just, again, it's Campy. It's a hard bug.
It's a different organism. We have to approach it differently.
Yes, you do. But I do think that we can do further reductions. We just need to understand our multi-hurdles a little bit better and really start to apply them in targets. What is our goal and that for that step beyond just reducing pathogens? One of my concerns too is shelf life. If we're using PAA, that's an organic acid that, may have a pKa around 4.5, a little bit higher, still acidic, but it's where our lactics are probably going to resume growth. And so we've done some shelf life studies where we've looked at PAA versus an inorganic acid and looked for those not just indicator plating, but also microbiome. So what is that
What else is
microbial
still there?
ecology? Yep. And so we saw, a shortened shelf life compared to the inorganic acid, which had a pKa of 1.9 versus a much, higher for PAA. But we also see with that, we saw that there was a more selectivity towards some of these shelf life organisms when using PAA. And so that's something too to consider, right? So we've got to start thinking not just pathogens too, which pathogens are very important. We want food safety, but we need to think shelf life too, because our products don't have a very long shelf life, right? We, out of the meats, we're probably one of the shorter shelf life, especially for tray packed foods. So we, anything that we can get an extra day maybe is very important, not just for us, but for the consumer as well.
And in terms of that, in your work, in terms of looking at, almost like as you're describing this, we need almost a rotate. Are you talking about a rotation system in terms of plant chemicals, or using things in conjunction so that you're getting kind of a two-step knockdown? So, or both? What do you think is kind of application of this research in terms of, in a poultry processing plant, next step? Okay, yes, everybody's using the same PAA everywhere. Your research is sort of fine-tuning it. Okay, we need to look at it from this approach. So rotation type of system, or almost a two-step kind of thing?
So less on rotation and more on a multi-hurdle, so a two-step process. So say if we decide in the chiller we're going to use PAA, well, let's in the post-chiller use maybe an inorganic acid or maybe use CPC. CPC is really good at, we saw that it had a differential effect between Typhimurium fanis. It actually reduced some of those key genes that regulate invasion. And so we did a skin invasion assay, and we saw that different effect and that response of the genetic component. So that's, adding more of those components, multi-hurdle. So you're getting a different type of mode of action throughout the facility. And then so we've got PAA in the chiller, the post-chiller, we have CPC, or maybe we've got an inorganic acid, and then we've got a part dip if we're talking about,
right,
tray pack system. Yep. Then you can go and add something else to that. Maybe a low concentration of acidified sodium chlorate or something else. But really, one of the points of the research I want to understand is how does it impact, and when we start adding that multi-hurdle approach, so we add one on top of the other, right? So if, say, we transfer salmonella and we've stressed it out with PAA, what's the effect of CPC after that? Well, what's the effect? Because there's not a lot of research to look at those effects. We're going to start in probably in vitro. We're going to have to, so we can see those effects without multiple other factors being considered. But then we'll go on to meat matrices. But that's multi-hurdle. But then also, if we see a differential effect between Salmonella and Campylobacter with some of these different antimicrobial interventions or even novel interventions, then we want to make sure that okay, your plant has a Campy problem. Well, I recommend this product at this sequence, and that'll help you get Campylobacter reduced. And you'll still have similar Salmonella reductions most likely, because we know Salmonella is a little bit easier to target, but we know that we can also effectively reduce Campy too. So kind of twofold for multi-hurdle, but also specific to pathogen of concern. And if you think even there's a serovar of concern, that's something hopefully that I want to look at too, is not just cocktails or individual, I want to look at those individuals as well. So is there a difference between Typhimurium and, Infantis, like I previously said, or is there a difference between Campylobacter jejuni and Campylobacter coli or Laurie? And this applies more than just the poultry industry too, because we do have similar uses in the other meat industry as well. I know the beef industry uses PA as well, and so does the pork. So that's just one of those things. But really, yes, trying to apply it differently, not just, I also want to apply physical after the in vitro assay, that physical component as well. So rinsing, or if we're talking about a scalding system, so temperature is also an important component to include, really trying to include those factors and understanding the response of the pathogen to those factors. Then hopefully we can say, you got a problem with this, here's the map that you need to use to get to it. Rather than just saying, oh, here's my product, I sell PAA.
Right.
Let's get you some PAA and it'll solve all your problems.
Right.
And I'm not saying it's a bad product.
No.
It's a great product and it has worked really well. But when we start to have a dependence on one product, we start to see selectivity. And that's something that we really need to be concerned about. It's not just selectivity, but tolerance buildup of certain microorganisms.
Absolutely. I think we put different interventions, and then we will control certain serovars, then that allows other serovars to become predominant. Both pre-harvest and, everything that we're doing is just selecting. And, it's probably how we got to where Infantis, 10 years ago, no one even talked about Infantis. It was, or maybe even less than 10, but, these serovars sort of emerge. So our control strategies are definitely influencing that ecology. We have just a few more minutes left since we focused a lot on the plant. And I know you have some background in terms of pre-harvest strategies, and that's another area that we look at. So are you doing any continuing work on that side, or is everything really focused for you on the plant, or are you continuing to look at nutrition and how that influences, the gut microbiome and these commensals that we're looking at in terms of Salmonella and Campy?
Yeah. So I do focus a lot on post-harvest in my current position, but one of my strong beliefs is that we can't only control it post-harvest. Like, there's only so much you can do on the back end. You have to be proactive about it as well. So at the pre-harvest level, I still have some goals and aspirations in that, and still help out with in-feed supplementation and how that impacts post-harvest food safety, or even looking at the microbial ecology. So recently just published a paper with Christy Sagerty looking at a botanical blend of an encapsulated botanical blend on necrotic enteritis model. And so what we were able to see there, which was kind of cool, is that you didn't necessarily see a lot of big differences in the microbiome, but we saw differences within the core microbiome over that study. And so, with the supplementation, we improved some of our other characteristics as well, but with that core microbiome, we started to have a more selectivity towards a normal microbiome. So some of those really good microorganisms like Verrucou microbiota, it's a very hard word to say, promise you. But
oh yeah, you look at some of those like ones where they show those big charts with all the colors, and you're like, what's the name of that bacteria? I can't even pronounce that.
I chose the wrong career for my pronunciation, but
and then they changed the name. We were just talking about that yesterday, how the names keep changing. And it's like, just when you're teaching students, or you're like, oh, by the way, I feel like we were even talking about Clostridium, and one of them has been reclassified. Like, it's not even, and I was like, I just can't, it's hard to keep up with that.
There was I noticed that the other day on Wikipedia, of all places, told me that a whole family was renamed, and I'm like, no, it's a whole phyla, not a whole family. A whole phyla was renamed.
Well, that's
a whole phyla. And I'm like, well, I wish they would have let the scientific community know about this.
Right.
And it is backed up. It's published paper. It is like, that's a little too much. But I'm also kind of interested in not just in feed supplementation, but welfare as well as stressors that the birds or animals might have to go through.
Right.
As we know, stress can be a big player on increasing.
Stress definitely increases salmonella shedding. Major.
Yes. And the pathogenicity. So, my old PI, Dr. Ricky, one of the things that he also has showed in his research is that pathogenicity, it would be increased during feed withdrawal. So it's very important for some of these key stress points. I partnered with Dr. Sammy Drudy at the University of Arkansas, wrote a seed grant. So hopefully that gets funded. We're waiting on that. But wanting to investigate really the impact of heat stress on the shelf life of poultry products. When we understand that there's going to be bacterial translocation, there's going to be, that increase in gut dysbiosis, that should all translate to post-harvest. And so we really want to see that or understand that, and then hopefully create a framework for the industry to work around that. So maybe we need to use different packaging techniques coupled with different antimicrobial products so that we can then prolong that shelf life when we know that these products are going to have an influx of overall bacteria and maybe even shelf life spoilage organisms. So that's something that him and I really wanted to focus on, as well as focusing on understanding salmonella establishment during that time and how that translate to our finished product.
So you're talking about heat stress from just prior to processing, like in the actual bird, correct? Or during processing?
Housing.
During housing.
No, during housing. Yep. So this
that last 3 to 4 days prior to processing. Or
day
even
20
earlier.
To day 42.
Day 20 to 42. So that's a big window.
That is a big window. And so we have two kind of models for this. We have a cyclic model, which we will probably see more in like states like Arkansas, or maybe the southern states. I will tell you, Mississippi did not cycle significantly. We normally held about a 5-degree difference between.
Right. It's just hot.
It's just hot.
It's just hot.
It's hot and humid. But you would see more of a constant heat like that we're proposing, and more of tropical regions as well. But really, we just want to understand, because global warming, no matter what you believe, climate change is happening.
Climate change is happening. It's just climate change. The earth goes through cycles. It's just part of it. And our industry, I think, very certainly will have to adapt. Like I was actually talking to someone in California last week, and like just the huge snows that they had, the melts, they've had farms flood, like things like that, like things that used to not be in a flood, all of those environmental kind of things that you didn't have to think about. And the farm has been unreachable, so a lot of things our industry will have to deal with, and heat stress definitely is probably going to be one of those. So.
Oh yeah. And I mean, we've dealt with it before. We do have very controlled, climate, but even with, using wind tunnel systems, you're still going to have some issues.
You can only cool them so much, like that. It has the capacity to only cool so much. And then we still have, parts of like, especially the turkey industry, you may still have curtain-sided barns, and they don't, necessarily have the same cooling strategies. So it's a lot to work out.
8 years ago, but when I, worked on the East Coast and interned on the East Coast, we still had open houses. Open-sided.
In Delaware.
Yep. Not even current, open-sided.
Yeah. They're probably reduced in number by now, but yes.
Significantly.
Yes.
But, even back then, so that was 2013, that was very surprising to even see that in 2013. So actually, that's 10 years ago. Wow, I am showing my age.
Well, I got a few years on you, so we're good. We won't age ourselves.
Yeah, no.
It's time for our famous three. A worldwide leader in animal nutrition, Adiceo's portfolio of products includes methionine, the full range of vitamins, enzymes, organic selenium, probiotics, mycotoxin management strategies, and palatability products. With such a diverse offering, Adiceo supports its customers with a broad range of expertise, tools, and services to help them maintain a competitive advantage. Adiceo, fueling predictable profits. To learn more, visit Adiceo at www.adiceo.com.
I think we've covered a lot of topics, and you definitely have some interesting research in the works. If you had one piece of advice to give, we have students often listen to us. So what kind of advice would you give a student that's interested in sort of food safety and processing? Where would you guide them?
So one of the things that was the most beneficial for me, so this is a great advice, I believe, in my own opinion, is when you have colleagues or fellow students doing research projects, take the time to help them on their big days. And it will give you so much experience. It helped me to branch more easily from pre-harvest to post-harvest and even establish my master's and doctoral research, experimental design, because I was able to see what they had done and why they had done it and how they had done it. That experience, you can't necessarily always understand from reading a paper when you're a first-year master's student, but you can understand when you see.
You can understand it when you're harvesting organs or oral gavaging salmonellas, and you understand how all of that works when you're part of the team. That's great advice. That's awesome. Well, Dana, thanks for the chat today, and thanks for joining us on the Poultry Podcast show. And it was a pleasure talking to you today.
Thank you so much, Karen. Thank you. It was a pleasure.